see. So we'll be getting started at 5 after in just a few minutes. Just a couple more minutes and then I'll introduce our panel and we can get going. All right, it's 5 after, so I'm going to say a few words about the format of today's event, and then we can go ahead and get started. Thank you all for coming to today's panel on reproducibility and neuroscience. We're very excited for this discussion, and it's my pleasure to have as our guests today, Verena Heiss, Guillaume Rousselet, and Dorothy Bishop. Russ Poldrick sends his apologies. He's not able to come today due to unforeseeable circumstances. But I think we'll still have a wonderful discussion with our three panelists. The way that today's event is formatted is that we're going to have a brief introduction from each of our speakers where they talk for a few minutes about who they are and how they came to be an advocate for reproducibility in neuroscience. Then we'll have a 25-minute discussion led by a few questions from myself. And then we're going to have 25 minutes of audience Q&A. And so for the audience Q&A, please feel free to type any questions that you've got in the Zoom chat as our speakers are discussing. And at the end, I'll be going through and selecting some of the questions. And at that point, you'll have the opportunity to turn on your camera and unmute your microphone and ask yourself if you would like, or I can ask for you. With that out of the way, I suppose it's time that we get started. Could we start with Guillaume? Would you be willing to tell us a bit about who you are, what your background is, and how you came to become interested in reproducibility?
Yeah, sure. Hi, everyone. So I'm a senior lecturer at the University of Glasgow, where I work mostly on visual perception and individual differences, aging. Well, I guess it used to be my main line of work. More and more, I feel like I work on methods and statistics. I'm drifting progressively. I find that more and more interesting. You know, if you ask me between spending time in the lab and opening an R notebook and starting to run simulations, I'll go for the simulations every time now. I feel like I have more control in that environment. How I came to work on these sort of things and try to promote open science and reproducibility and so on. I guess I've been very lucky starting early on during my PhD, then my postdoc, and then my first job at Glasgow Uni was to work with a lot of people who did things very differently from the rest. So when I started my PhD, I was working on visual perception of natural scenes and doing EEG measurements. And the way I was introduced to the field was like, well, if you read the papers, that's how people do things. Here in the lab, we do things very differently. So there was that attitude that you could be in a situation where the majority of people do things that one way, but it's not necessarily the best way to do it. And I think that was the best start for me. When everything started really to click was when I joined Twitter a few years ago. And I joined that amazing community of really open-minded people pushing for reform. And I've discovered so much through that. But I think if I have one big debt, it's to the open science Twitter community. I've just learned so much that was not available anywhere else. So, and it's still my main source of information, really. So if I have just one recommendation is to go on Twitter and start following great people like Dorothy, for instance. That's actually, Dorothy was, I think, with the second or third person I started to follow on Twitter back in the days. And it all started like that, you know, you start following recommendations, papers and so on, and it just turns into something else. But it is a long and slow process, though, I must say. So I think I'll stop here for the introduction.
Awesome. Well, we're really glad to have you. And on that note, maybe Dorothy could introduce herself and tell us a bit about herself.
After such a lovely introduction already. So yes, I'm a neuropsychologist by training, and with an interest in children's language disorders, genetics. I have also done some EEG work in my time, and it veers into psycholinguistics. So it's a lovely area in the sense that it's got a lot of theoretical interest, there's lots not known, and it also has a practical slant to it in the sense that you may be able to develop interventions and so on. But studying quite a lot of different areas in the course of a very long career, I became more and more aware of problems in not just my field, but in psychology and in related fields of medicine. Also influenced by the fact that I was a journal editor for a good 8 years. I was editor of Journal of Child Psychology and Psychiatry. And so I got aware of what happens in the publishing system. And I guess what I was, by this time, I also have a debt to social media. It's interesting that Guillaume says that, because I actually got into Twitter from blogging, or the other way around. I think, yeah, I found Twitter via Sophie Scott, who was another famous Twitterer. And I just noticed that there were interesting things happening on Twitter. And then I found that I wanted to say things in more than the allowed number of characters. So I started a blog. And I found that my blog was more and more really complex. It was me giving me a chance to have a good moan about things that I was concerned about. And a lot of them had to do with reproducibility, had to do with things like publication bias, the fact that we tend to only publish positive findings, and that journals will often accept a much lower methodological standard for something that looks very sexy than a much better designed study that doesn't have such an exciting result. And I think I gradually realized that has a huge negative impact on science. I've got very, very interested in the other thing that biases the results, which is within a study when people analyze things many different ways, looking for the significant p-value, so-called p-hacking. And I, like Guillaume, again, I'm very interested in using simulations to understand statistics. And I have been doing that for a very long time. I'm really quite geeky. And I found that I would sometimes quite shock myself at the extent to which you could get spurious positive results if you were very flexible in your analysis, or if you had lots of different things you could look at. So that was the other thing that concerned me. And the tendency for people to sort of invent a story about the data only after seeing it, so-called harking, hypothesizing after results are known. And crossing different areas, it was interesting because some fields seemed quite aware of the problems, but didn't necessarily do anything about it. And then other fields, like the EEG field, which was more inhabited quite often by people who had strong backgrounds in engineering and were really very competent computationally and so on, but nevertheless, they didn't really understand the statistics very well. So I would have journal editors encouraging me to just sort of look at my data every which way until I found something, which is really not helpful. And if you do simulations, you start to see how dangerous that is for bringing out type 1 errors. So then I suppose the watershed for me was, I think it was about 2015, I was invited to chair a symposium that the Academy of Medical Sciences was running on reproducibility and replication, I think it was, in biomedical sciences. They did that jointly with Wellcome Trust, MRC and BBSRC, and it was very interesting to see that these three funders were getting very twitchy and concerned that biomedical science seemed to be full of non-replicable findings. And that was fascinating because they invited people from industry, they invited people from other disciplines. I learned about aspects of poor reproducibility that don't apply to psychology, but were very serious, like people not being sure what their reagents actually were, contamination of cell lines, things like that affecting research. And this was all discussed very full and frankly. But the question was then what do you do about it, which is the topic here today, I know. And we ended up concluding that it wasn't a single problem. And it was in part a problem related to how scientists did science, but that this was also very heavily influenced by top-down things like incentive structures in science, how the journals behaved in terms of what articles were accepted, how funders behaved in terms of what research was funded, and that you needed solutions really at the structural level. But you also needed better training of people who just didn't appreciate some of these problems with things like p-hacking. And the funny thing was that the Academy of Medical Sciences, wonderful organization which organized this and did a really great job, but they do move at a rather slow speed. And at the end, we had these recommendations and they were very slowly moving towards sort of formalizing the recommendations. And with Marcus Manafo and Chris Chambers, we'd had a discussion about the idea that it would be great to do some training. And an opportunity just came up that BBSRC, who had been one of the funders of this meeting, were advertised some funding, not necessarily for training, but some sort of rather weird, funny little bit of money. And I thought, oh, we could use that to do some training. And so we applied for it and got this funding to run a course, which we ran for 4 years annually on advanced methods in reproducibility. And indeed, Verena was, I think, in one of our first years as a student. And that has been marvelous in bringing together different people from different fields, mostly psychology, but also other areas of biomedical science, and even veering off into other disciplines altogether. We sometimes get a brave economist or something, people coming from really quite far afield, which is great because you can then see how these problems either are similar across areas or manifest differently. So I've become more and more, like Guillaume, more and more, I mean, I find now most of the talks I'm asked to give are not on child language disorders, but are actually on reproducibility, although I do also still work on child language disorders. But I think I feel very passionately, this is a problem we can fix and we need to fix, because I think we're spending a huge amount of money on research that could be better. And also people's careers are tied up in this. People are being often encouraged to do science in a way that I think is not leading to forward progress, but it's very much they're being encouraged to think they've got to make some big splash with one huge study that gets into a very sexy journal. And that's not how science should work. It should be cumulative. People should be scholarly in their approach to the prior literature, and we should be able to build a sort of solid edifice of evidence. So I'm hoping that, I think things are changing and in the right direction, and the fact that the funders are keen to move and not waste their money is a great thing. But I think that's probably given you an idea of where I'm coming from. So I'll stop and let Verena take over.
Yeah, thank you for that. And I'm glad that you brought up the dichotomy between institutional factors and individual factors with things like the publication bias or file drawer problem. We'll definitely be coming back to that.
Yeah.
But yeah, now we're happy to hear from Verena about where she's coming from.
Yeah, absolutely. Well, first of all, thank you so much for having me. I think it's absolutely brilliant to be on this panel. It's brilliant to be back in Oxford in some way. It's wonderful to see so many familiar faces or familiar names, at least on the participant list. And so about me, I'm not a psychologist here. I'm a molecular biologist by training, and I've actually done quite a number of different things over the last couple of years. So I've started in molecular biology, then decided that I wanted to study the human brain. So I've spent a number of years doing human neuroimaging. And then I realized, well, actually the samples that I want to look at need to be much bigger, because I wanted to link up genetics with neuroimaging. And that's how I started to become really interested in epidemiology. So I did another master's in that too. So I guess what I'm trying to say is I've seen a lot of different fields in biomedical sciences, and that actually helps me with the research that I'm currently doing. So I'm now a research fellow at an Institute for Advanced Study in Germany, and I'm super grateful to them. They're called Hanse Wissenschaftskolleg, because they decided to give me funding to do a relatively crazy project, which is to look at reproducibility and open science in different fields of biomedical sciences, to try and understand in how far are the issues that we're facing similar or different, and in how far can we use solutions that have been developed, for example, for areas such as epidemiology, and use them in other areas such as bench research, for example. So this is what I'm currently doing. It sounds like a very crazy project, but I'm really enjoying it. I'm really enjoying that I'm having so much time to actually read and read the literature, which is something that I can always just recommend to anyone. And yeah, my reproducibility journey is I thought quite a bit about this when we got the brief to say something about this for the panel here. And I think, yes, it's obviously thanks to Dorothy that I'm here because of the training course. For me, it started a bit earlier with this sort of feeling that there seems to be a bit of a mismatch between really what's good for science. And what's good for science is having reliable results, having things like collaborative research, but also getting research into the clinic, right? This is what I am really passionate about. I'm doing research into Alzheimer's disease. So I really care about my results being useful in the clinic at some point down the line, maybe not next year, but maybe in 5 or 10 years time. And that's, what's good for science. But really what's good for individual scientists is very different from that, right? It's getting like Dorothy said, it's getting a sexy result in a potentially high impact factor journal. And those sexy results, we all know, tend to be false positives in a lot of cases. And so rather than really thinking about the reliability of our research, I think we just tend to think about really good stories. And so I feel like, this mismatch between what's good for science and what's good for scientists basically put me at a crossroads. I felt like, okay, if this is not the kind of system that I want to work in, and this is not how I want to work personally. And I think in some ways what saved me was the course that Dorothy just described, the course on reproducible, I think it's called Advanced Methods for Reproducible Science. And we did this I did this in 2017. And I think what struck me with this course was not just the fact that I could do my own research differently, but also that there is a whole community of people out there who are thinking about the world in a different way, and who are thinking about academic research and changing the way that we work, really. And so I think following on from that, we started to do some training and run some training in Oxford. We ran a number of Oxford reproducibility schools. I got very involved in organizing or co-organizing the Berlin-Oxford Summer School on Open Research together with colleagues at the Berlin Institute of Health. And then one of the things that also happened at the same time was colleagues in experimental psychology starting the Reproducibility Journal Club. And there were all sorts of activities going on around reproducibility where we realized, hang on, we need to get together here in Oxford because there seem to be lots of people who are interested in this. And I also realized in some ways it's not just about training, right? We do need the right training, but we also need the right infrastructure. We need to change incentives. And in some ways, we also need the meta research to show that, the solutions that we propose actually do work. And this is also what Dorothy talked about earlier. We need to think about the scientific ecosystem and think about how each of the different players can play their role, institutions, societies, funders, publishers, industry. Let's get together and think about what a really good system would look like. And so I'm very passionate about getting people together, about getting the community together, and that's one of the things that we I think we're quite successful at doing here in Oxford, where we've now got together a wonderful group of people that's now called Reproducible Research Oxford. For those of you who haven't heard of ROX yet, I guess my sort of the best kind of advice that I could give you is go to the website of Reproducible Research Oxford. Check out what's currently going on, what the events are, what resources are out there, and also just get on the mailing list. So I have to admit, I'm not on Twitter. I hate social media, and it's always a bit of a pain for me to hear that it's all about Twitter. I totally realize that it's a great resource for everyone, but I would also encourage people not to think that you have to be on Twitter to work in this space. So yeah, that's me in a nutshell.
Thank you so much. And I posted the link to Rox's webpage in the Zoom chat, so any interested viewers can go there, sign up for the mailing list, read about it, that kind of thing. So before we delve into the more practical advice section of today's panel, I wanted to start off with a general question, which I think is something that all of you have alluded to in different ways. And to sort of motivate this question a little more, I'm going to read a short section from a press release last year that the British Neuroscience Association published that I think summed this up very nice and succinctly. They wrote that modern science incentivizes the publication of as many papers as possible in journals of high impact. This inevitably creates a temptation to rush, to cut corners, and to articulate impactful stories that may, to at least some degree, distort the true nature of findings. And yet, also there are simultaneous challenges regarding individual researchers' statistical literacy and data reporting. And so I wanted to ask if each of you could talk for just a couple of minutes about, from your standpoint, the relationship between the structural and individual barriers to reproducibility in neuroscience. If anyone has really strong thoughts, they can jump in first, or if not, we can, okay.
Well, from that code, maybe I can start, because I'm editor in 2 neuroscience journals, and that's very much my experience. And when I get a paper to edit, the first thing I do, I'll scan the paper very quickly, and the first thing I look at are the figures, and the quality of the figures correlates really strongly with the quality of the research and the inferences. And my main thing is, if I get a paper full of bar graphs, I know it's going to be desk rejected, or an invitation to resubmit once we can have a proper discussion, once we can see the data. And the next thing I will do is check if the data have been shared. And those two factors really correlate super. I could just look at that and I'll be 90% correct without reading the rest of the paper. And then I look at the results section. And what do we find in the results section? Most of the time, it's like we're in zombie land. We have scientists who may have a really nice, complex background story. They put together a lot of different sources of information to craft a nice experiment. The experimental design might be very nice. When it comes to the analysis, it's zombie land. It's just people have been doing some tests, probably going in SPSS, clicking on the button, then they look at the p-value, and then they make this binary decision whether it's inferior or not to 0.05. And there's it's a complete different world. You might read a well-crafted introduction that is all rich and so on, and it comes to the result section, there's no science there anymore. It's pure recipes, and there's no nuance, and everything that is discussed then is all completely binary. Or they found this and they found that, but it's not about numerical values. It's about a binary outcome, whether it was significant or not. And that, to me, is the most negative aspect of most of the research I read. It's just like there's most of the outcomes are just unusable because the data are not shared. There's no justification for anything that's been done. By default, it's going to be some random test like a t-test or ANOVA. And there's no discussion of any of that. People just follow recipes. And that's why I think we have a major issue in reproducibility, is that people follow those recipes without thinking. And then when it comes to assessing whether something can be replicated or not, it's only about the binary outcomes, significant or not, as opposed to precise measurements. And it's a typical statistical fallacy. If you have an effect that is significant and other than is not, it doesn't mean that the two actually disagree with each other. So for me, that's the main problem I see in most of the stuff that is published, this complete discrepancy between how people think about their research and how they actually carry it out, and how we can use the output. And for most of the outputs I see, it's utterly useless. It's a complete waste of time and money without having proper illustrations, proper data sharing and justification.
I agree with much of what you say, but I would say we do need to think about the motivating factors. So my shocking story, I suppose, but it probably won't shock anybody here, was somebody who I knew who was at an institute, who was actually a professorial level at an institution which was in financial difficulties and cutting down. And so this person had been threatened with possible redundancy, and was talking to me about this. And she said that, well, she had a paper under consideration in PNAS. And it was very clear that, in the interviews she'd been having with various higher ups in her institution, that if this paper was accepted in PNAS, she probably would be kept on. If it wasn't, she probably wouldn't. Now this, just imagine how corrupting that potentially is, apart from being inhumane. If you're in that situation and then the referees say, well, you just got to do this one more experiment, and the temptation to fudge things, to drop data that don't fit, if your career is on the line, depending on this one paper, this is wrong in the sense that what should determine publication of that paper is whether a good question has been identified and you have done a really good job with a methodologically tight, well-designed study that will give us a clear answer as to what's going on, the sort of thing that Guillaume's talking about. So my feeling is that we should be evaluating people in terms of the rigor of their methods, their scholarship, whether they're asking questions that people actually want to know the answers to, and not by the results that they come up with, which then may or may not get published in one of these sexy journals. But I think I don't think it's so much about quantity of papers, although I think that's more the case in some disciplines and in the US. In the UK, there shouldn't really be pressure on quantity of papers. The REF is always blamed for this, but the REF actually has the opposite pressures on people, because it only allows you to submit a finite, quite small number of papers. So you would be sensible to write fewer, much more meaty, impressive papers than spin out lots and lots of little ones. But people somehow seem to feel that they've got to spin out lots and lots of papers. But I think it's more that what you're judged on is so much on the results rather than whether the science was well done. And there's this, again, this bias towards positive results. And of course, we all want exciting, positive things. And if you do a vaccine trial, we want it to work. But it's also very important to know what doesn't work and to have a study done properly so that if it doesn't work, we know, okay, that's one we can tick off the list. And other people don't spend time trying to develop that vaccine if it wasn't really effective. So it's this, I think it's this pressure on people to produce the big glittering result. It has been very, very bad. And it really, I mean, I've given talks where I've said to people, you should be doing this, you should be open data. They say, well, if I do that, I've got a mortgage, I've got a family to feed. You think, what on earth is going on if we've got to a position where people do feel, just as Verena says, that there's a conflict between what you need to do to survive in this ecosystem and what is good science. That needs to be realigned. It needs dramatically to be realigned so that people are not trying to fight against a system that's forcing them to do things that they don't want to do, but which are actually dishonest.
Yeah, I'm also happy to chip in if you want. I think one of I totally agree with what Guillaume and Dorothy have said so far, obviously. I think one of the things we haven't mentioned yet is this factor that we all have to be jack of all trades nowadays. So I think we're supposed to know a lot of different things. So, we should know the biology, we should know about statistics, we should know about machine learning tools, what have you. And then we also, I guess most of us do some sort of coding. So we have to, in some ways, become software engineers too. And I just wonder whether this idea that we have to be experts in everything is also not helping very much. I think this, one of the issues that I think we're facing is that science or the different disciplines become very complex, and the data sets that we work with become more and more complex. And I just feel like, we don't think about collaborative science enough. I think we should be much more collaborative in our approaches. We should have people with different, outlooks, different disciplines working together, rather than having this super competitive system where everyone just fights for their own survival. And I think, team science is one of those issues that might actually fix some of the issues that we've talked about.
Yeah. I think that's so important to recognize that it's really unrealistic for all of us to be experts in everything that we have to do to get a paper to publication with rigorous methods and rigorous analysis and reporting. Kind of building on that a little bit, I have a question for Guillaume. So we know that you've advocated extensively for increased transparency in data reporting, and you've even written in one of your blog posts that you've reached a stage where your papers are free of p-values. And so I was hoping you could talk about what you mean by that, and if you have any advice you could give a researcher who feels like they don't have a solid foundation in statistics or data reporting.
Yeah, that's a question I get asked quite often. But I change my mind every day pretty much, comes and goes, including my position about p-values. But it's a really tricky one. I feel like what Verena was saying is very much true. Like this idea that one person should be able to do everything in a study is completely bogus. We know it just doesn't work. In medical research, people team up, they always have statisticians, and the statisticians are involved ideally at the start when the experiment is being designed, because the two go hand in hand. You know, the statistic is not something that you sprinkle on the data at the end, like icing on a cake. It's something that's there at the very start. And the two, I realized that when I had a wonderful collaborator, Cyril Pernet, and when we started designing experiments together, we went to the whiteboard and starting to make drawings of what the figures would look like, how we would analyze the data, and then we were going back to the design, because as through that process, we realized, well, actually, we want to ask a different question. And you need to realize that statistics, it's not like applying statistical test to get a p-value. Each test is asking a different question. It gives you a different point of view on your data. And things, it's not like doing parametric versus non-parametric. They all ask very different questions. So you need to get into that follow that idea, and then you realize that there's a lot of flexibility. There's a lot of cool questions you can ask, and it's not just a matter of following recipes. So where do you start? I don't know. There's quite a few references I can recommend to get started. You need to read a lot. You need, if you really want to understand statistics, you need to have a scholarship of statistics, and that means spending the same amount of time and probably more reading about it, engaging, learning to program. And there's plenty of tutorials nowadays. So if you, I would advise learning R or Python, and in your field of expertise, that you'll find a lot of people are developing lots of tutorials and cool resources that are really useful, where you can download a data set and you'll be walked through different steps, and you can start thinking about all your different options and so on. But it's going to be painful. There's just no magic recipe. So many times people ask for a quick advice on how do I handle that. And it's like, hold on. I mean, it depends on so many things. What's the structure of the data? What do you want to ask? If you do that test, you ask that question. If you use that other test you had in mind, you're asking a different question, which is the question you're interested in. So it takes a lot of time, and there's no quick fix. And that's what I teach. I just finished teaching a level 4 option for undergraduate students at the University of Glasgow on using the Bootstrap. And that's the main thing I teach them, is that even with a flexible technique like the Bootstrap, you can ask a lot of questions. And I use simple example. You want to compare two groups. Well, there's 10 different ways to have a look at that. There's plenty of different graphical representation, different statistical tests you can do on that, and they all mean different things. So what do you want to know? And then they start scratching their head and they realize, well, actually, there are plenty of options, and the data are much richer than you would think if you were just doing a t-test. So I think scholarship is the key, but it's not easy. I have some references on my blog, for instance, of articles I recommend. But if people want to get in touch, they're working on something specific, maybe they want to have some advice. I'm not a specialist in everything, obviously, but the best place I know to get advice on stats is on Twitter. You can ask some of the leading biostatisticians on the planet directly, and I had amazing discussion about some of them. There's some really, really clever people out there.
I'm a little worried, though, that, I agree with Guillaume, and in fact, we've just, a small group of us in Oxford have just finished doing a book club where we're working through a complicated stats book by McElreath, and it's great. Yeah, it's a wonderful book, but we're all saying this is a killer. We thought we'd do a chapter a week. We then found we were doing a chapter every, sort of a third of a chapter a week, because it was so hard, and we would have given up if we weren't all doing it as a group. And, I think what worries me is, and we discussed this in the group, is that if you're not careful, people are turned off because it just all seems too hard. And what we used to say, this happened very much with our course, which was a week-long course where we tried to, cover lots of stuff, not just stats, but experimental design and open data, using, sharing code, doing systematic reviews, working collaboratively, and, but at the end of the week, people were saying, this is great, but they were all shell-shocked. And we had to sort of back off a bit and say, it doesn't matter if you just do one of these things to start with, do what you can, take it one step at a time. But if you want to make your data open, don't, you just do a little bit of that to start with, but start working that way. Don't think you've got to suddenly transform into somebody who's doing everything, because you'll drown, I think, if you do. So while I agree with Guillaume, and it is tough, and it's rewarding but tough, and one has to think hard about what one's doing, I think it's more, you have to have the mindset to know how it is, and then you have to sort of, it's like, I suppose it's like, if you wanted to climb Everest, you don't just sort of get up and climb Everest. You train, and you do a little bit here and a little bit there, and you slowly make progress to that goal. And I think that's how you'd have to think about it. You have to have the goal in mind, but not try and do everything.
I mean, one of the structural issues that we see here very clearly as well is that we're not being taught those things in our undergrads, in our graduate studies. So why is it something that we have to learn on the side in some way? So I always wonder, why don't we get taught research methods in a much more fundamental way? At least in biology, that was never part of what I was taught. And I think the first time that I realized, how much knowledge I still need was when I thought, maybe I should do this epidemiology master's. And that actually completely changed my outlook on research. And I was like, wow, this is the first time that I'm being taught on how to do research, how the process of doing research works, what are biases and all these kind of things. And you might think, well, that sounds ridiculous. Like, why does it take you, years? It was after my PhD that I did this master's, right? So I think teaching research methods is really fundamental, and is something that we should integrate so much better in any kind of course that we're running.
Yeah, I think that that is really important advice. I know that I did my undergraduate degree in psychology, and when I was studying that, it was after the reproducibility crisis had reached its peak in the discipline of psychology. And so my university required a research methods course, and that's been invaluable to me. And I also think it's really telling that even on this panel, all three of you have talked about how at some inflection point you had to make the transition to start focusing more on reproducibility. And as Guillaume and Dorothy said, now that's a major area of research. I think it's quite telling that delving into this to such a level of expertise has really become a part of your professional trajectory. It's not something which is very easy to just become an expert in just a weekend or anything like that.
No, but at the same time, I feel strongly that I'm still interested in the area of language disorders, brain lateralization, and those things. So what I try to do is I wouldn't want to just write about reproducibility. So what I try and do is lead by example and try and implement things, like open data, like having reproducible code, but with each and doing pre-registrations and registered reports, which I don't know how many of this audience are familiar with, but the idea that you are very upfront about your plans and your analysis, and with a registered report you can get that peer reviewed before you collect the data. So you're decoupling the evaluation of your work from the results. You're getting it evaluated in terms of whether it's a good study in principle. But it's hard, but it's very, very satisfying. But I do try and, I haven't done it all at once. Gradually, I feel my work is getting a little bit better with these different things being implemented. And the more you do them, the more it becomes second nature.
Yeah, another path to follow. So I'm working on a registered report at the moment, one of my first one, and it's awfully difficult.
Yeah.
It's you want to start to make predictions and plan everything ahead, and you realize you have no clue about half of the things. But it's a good exercise. The other path to follow is to be honest, but it's extremely difficult to be honest in this business, is to say, well, actually, my research is exploratory. And I'm going to publish my data sets with my code, and I'm going to make some lovely graphs. And I'm not going to have inferential statistics, p-values, and so on. I'm just going to describe things and trying to come up with cool new hypotheses to test in future experiment, and just be honest. But it's very difficult. There's only one journal on the planet that would offer, that has an exploratory report format at Cortex. When I have the time, I want to push that as a new format at the European Journal of Neuroscience, where I'm editor, because I really feel strongly about that. I want to give the opportunity of people to be honest. But nowadays you can't, it's like even in papers where people are honest and then say, that's our main stuff, we did our inferential statistics, and then they say we also had an exploratory analysis, and then they still have p-values, and you wonder what happened there. So it's back to zombie land. It's like, it seems that in neuroscience and psychology you cannot talk about a single result without adding a p-value at the end, and it just makes no sense. So we need to give the opportunity for people to be honest and to clearly distinguish what is exploratory from what is confirmatory. That would be a fantastic step. So I don't think the solution is just to go, registered reports. I think we need to give an opportunity for people to explore their data. Because in my field, in vision research, the stuff that got the Nobel Prize, the work from Hubel and Wiesel, was entirely exploratory. You look at their papers that gave them the Nobel Prize, there's not a single p-value, there's not a single statistical test, actually. It was careful, detailed observation based on some technical innovations and months spent in the lab making those careful measurements, which they describe, and most of the descriptions are hand drawings. And we learned so much. They completely changed the neuroscience in general and how we study perception. P-values were not needed for that job. But nowadays, they could not publish those papers because they don't have p-values. There's no t-test. So it's like an editor would just say, what is that? You need to put a t-test in there.
Yeah, I think that's a hugely important point. And I think you said that Cortex is the journal that currently offers a venue for this kind of exploratory work.
Oh, sorry. I was reading the chat. There's some interesting questions. Sorry, can you repeat?
Yeah, I said I was confirming you said it's Cortex that has that exploratory report format.
Mm-hmm. With very little uptake, yes, because people don't want to put their stuff there. Obviously,
Okay.
if people are honest and you have the option registered report versus exploratory report, the traditional format should not exist anymore. It should be banned. Right, the mixed bag of let's just throw everything into one type of paper, they shouldn't exist. Once you have journals offering registered report and exploratory report, they should get rid of the standard type of paper. That would be funny. I can't wait to be in a journal where that happens. Where everybody, if you submit a standard article, you're being forwarded to exploratory report and see what people say.
So to bring Verena in here, I was wondering, so we know that you're the early career researcher representative for BNA's Credibility Advisory Board. And I was hoping that in this discussion about all of the challenges that that all researchers face, from your point of view, are there any unique challenges that early career researchers face in improving the reproducibility of their research and advocating for reproducibility?
Yeah, sure. I mean, I guess the biggest one is that we don't have any sort of security, right? We don't have any job security. So I think that's sort of the biggest issue that many of us face, because when you're deciding to become an advocate, then basically what you say is, well, okay, I'll spend, maybe still most of my time on my research, but I'll also spend a lot of time on advocating. And sometimes that balance gets completely lost and I'm probably a good example of that. And so the question is like, how do you continue to have a job in academia if you don't publish as many papers as you could in your research field, but also if you're starting to do research that's slightly weird, if that makes sense, right? I mean, meta research, we don't really have that many outlets for meta research articles, although they are becoming more and more. So I think one of the biggest issues that, as always, we don't really have a lot of job security, and basically the kind of activism or lobbyism work that we're not doing doesn't get rewarded in any sense, right? On the other hand, I think I would actually like to say that for a lot of us, things have worked out quite well. So I think we have a lot of people in our community who have got jobs, the next postdoc position or whatever, they got hired specifically because they already have some skills in this area. I think reproducibility and open science skills, they are basically the future. And I think a lot of people are realizing this. A lot of PIs are realizing that this is some knowledge that they have to either keep in their labs or get into their labs. So I think a lot of us are actually quite at an advantage to some degree. But to me, one of the things that I keep telling people is that I don't care about impact factor anymore when I publish my papers. What I care about is whether I go with an ethical publisher or not. And for me, ethical publishing means I'm going to a non-commercial publisher. And so I've decided that for my own research and for my own sanity, I don't want to worry about impact factor of the journal that I publish in. And obviously, I have absolutely no clue whether that's going to work out, whether that's going to damage my career. But in some ways, I just feel like, there is a certain way in which I want to do science. And if I can't be successful, then fine, then I'll do something else. I think as one of the things that I keep telling, or that we talk about quite a lot in our community, is how fixed are you on a career in academia? If you're super fixed on this, then maybe the sort of traditional way, getting a paper into Nature or Science or whatever, will be the way to go for you, because this is in the short term, this is going to keep you successful. But in the long term, is this going to be good for science? I don't think so. Is this going to be good for your own mental health? I don't think so either. So I think in order to keep myself sane, I'm going to try and do things my way, and I'll just see if I'll be successful in that way.
I think that's a really important attitude to take towards all of this. And being an early career researcher myself, a graduate student, I've heard a lot of conversations from people who are increasingly keeping their options open to going for a position in industry. And so I think there's a lot of power in doing science on your own terms. I think it's better for science and better for our own mental health. Did you want to jump in, Guillaume?
Yeah, but the one thing, the one thing that really helps too is your supervisor. If you're a PhD student or a postdoc, your supervisor will dramatically change your outlook and your options. And unfortunately, I can understand that some people are stuck in labs. I sometimes get emails out of the blue from PhD students who say, well, I've read your blog and I'd like to do things your way, but what can I do? My supervisor doesn't want me to do that. And I don't have a good answer for that. That's the one thing, is that we all scientists, we should be open-minded, and whether you're a student or not doesn't matter. You can try to convince your supervisor to do things differently. I know sometimes it's going to be a tough battle, but there's more and more resources available, and it looks like in Oxford you have a nice community, and you can try to talk to other people. It's yeah, I don't have an easy answer to that, but I know some people will have an easier ride than others.
Yeah, can I comment on that directly? I think that's a super important point, right? So some of us are stuck with supervisors that are not necessarily super helpful in this way, but I think we need to, I think the way I approach this is we need to work with people, right? And we need to convince them that what we're doing is in some ways the right thing to do, but also it helps. For example, when you publish your data, when you publish open access, we do have a bit of evidence that shows that your papers are cited more, and this is something that your supervisor might care about. So just having the conversation with your supervisor to say, hang on, this is actually helping you as well. It's not just for me. I'm not just doing this because I like it. It's actually good for you. It's good for science in some way. It's going to further the field. So I think aligning your agenda with your supervisors is very helpful. And I think also in some ways, as early career researchers, we should pick the supervisors we want, right? I think that's really hard for PhD students. It gets a lot easier when you're thinking about your first postdoc, because at that stage, you're probably going to pick the person you want to work with, and so work with someone who works in a way that you agree with. And so, in some ways, I think we have a duty to be good citizens in the way that we also promote the supervisors who are doing really good work, and we just keep working with them and hope to get papers out for them and with them so that they can stay in academia as well.
Yeah, I think those are all fantastic points. And I want to ask one last question of the panel before we move on to the audience Q&A. Starting with Dorothy specifically, I know that you've written quite a bit about some of the challenges facing specific techniques in neuroscience, like your presentations on EEG and fMRI specifically. And so while there's a lot of discussion about the broadly applicable advice for improving reproducibility of our work, like registered reports and proper analyses, there's not as much discussion about methodology-specific challenges. And so we were wondering if you could talk a little bit about how researchers can identify specific challenges for reproducibility faced by their own study methodologies.
Yes, I mean, I should start by saying I'm not an expert in fMRI at all. I have worked with Kate Watkins, who is, but EEG I have, again, I don't know about being an expert, but I have some experience of. And I think that does have unique problems of its own, which have to do with the fact that which perhaps to some extent also are shared by fMRI, and has to do with the fact that you've got too much data, really. We did quite simple stuff with 64 channels and nothing too fancy with that. But even so, you've got this problem of analytic flexibility, because if you think about p-hacking, the most obvious source of p-hacking is when you have lots of dependent measures, like you're doing a trial for some drug and you've got 30 outcome measures and then you just pick the ones that seem to show an effect. But the other way in which you can p-hack is to just be very flexible in how you, for example, pick a time interval that you're going to look at, or pick a frequency band that you're going to look at if you're doing frequency analysis, or pick a particular electrode or a combination of electrodes. So that is a big reason that people are often really unaware of. And very often they do this overt thing. I mean, I was interested in ERP, event-related potential components, where you are actually picking a time window and just measuring the size of a peak in a time window. And people would often look at the data and select the time window on the basis of looking at the data, not realizing that that's a sort of double dipping, because that's building random error into where you put your window, but increasing the chances of getting a so-called significant result that way. So those sorts of things people in that field, I think, have as a problem. And then you have these weird designs where people would be using analysis of variance, which would be 3-way, 4-way, or even 5. I've even seen 6-way analysis of variance, without realizing that if you then are interested in any significant interaction, you should be correcting for the number of comparisons you're doing. People, including me, assume, for many years, I just assumed that ANOVA took care of the fact that you were having these multiple terms to look at. What ANOVA takes care of is the fact that there's perhaps many levels of a factor, and that is automatically adjusted for. It is not automatically adjusted for that if you do 3-way ANOVA, I think you might end up with 15 terms in terms of all or it might be a 4-way, I can never remember which is which, but you're essentially potentially able to see all sorts of different effects, and you need to not just take the view that you're interested in anything that reaches less than the magic 0.05. That way false positives lie. So those two things, particularly in the field of EEG, I think it's the analytic flexibility in many domains in terms of space and time, coupled with these complex sorts of analysis that are going to give you false positives, coupled with the fact that nobody ever tries to replicate things directly, or even if you do, then people say that's not a very interesting result, it's a replication. That's what happened to me. I was outraged because I was very concerned that nothing in this field, particularly with kids with language disorders, ever replicated, because everybody did it differently. And you could see that they all did it differently because they've been p-hacking. So some person would pick one window, another person would pick another window, some people one electrode or another electrode. And clearly people have been mining through their data to find something that reached the magic level. And so you had a literature that looked as if it was consistent because all these people had done studies showing an effect of something. But when you looked at it in detail, they were inconsistent because they were all doing it differently. And so I was really pleased when I actually got a result that replicated somebody else's finding, something I can at last believe, only to be told by a journal that this is of no interest because it's merely a replication. So I think it's a field where if you're so focused on novelty that you do not regard replication as of interest, you're really in big trouble because you don't have something cumulative then. You have a literature with little spotty bits of this and that all over the place. So that I think is the particular problem that's worse in that field than in others, partly because of the large amount of data that you've got. The fact that the designs tend to lend themselves to this sort of multi-factor ANOVA.
Yeah, I know that a lot of specific techniques face challenges like this, like EEG, and I'm sure many of us have heard of the dead salmon in the MRI scanner and how if you don't correct for the number of voxel-wise comparisons, you can get a lot of type 1 errors.
Yeah.
I was wondering if any of you have advice on where an early career researcher who is using a particular technique, say cell culture or EEG, where we should be looking to find advice for how to overcome these issues specific to our own methodologies.
I think if you're looking at statistical methods, then you should just be moving towards simulation. People often come up to me and say, well, I'm using some fancy multi-level modeling or something. Do I need to worry about this? I say, I don't know, but simulate it. Simulate null data, run it through an analysis. Do you get lots of positive results? The data's null. Therefore, if you're getting a lot of positive results coming out, that something is wrong and you should be doing some correction. So I don't know about cell culture. I mean, that's too far from my area of expertise. But I think where you're talking about analysis and how analytic decisions might lead to either missing true effects if you're maybe underpowered, or seeing false positive effects, there's no better way to learn than to simulate data. And that's not all that hard. I mean, it's not trivial, but as Guillaume said, if you learn R or Python, it's to me, it's a sort of, that is the one thing I would recommend everybody try and do, rather than just finding a textbook that gives you another rule to follow.
No, and most of the textbooks are completely rubbish. In statistics, the number of textbooks who get the definition of p-values and confidence interval wrong is just astounding. Again, go on Twitter and follow people in your field. There's a strong correlation. The probability that someone is on Twitter and will also have recommendations or may have been working on a cool tutorial to do a power analysis is quite high. So give it a shot. You never know, and it's quite a friendly community. And if you ask around, people will give you some advice. I mean, that's where I find most of the papers I read nowadays on the method and stats. It's through Twitter.
So I've tried to post a couple of things in the chat. I think for MRI, the best thing I can tell you to do is go to the Open Neuroimaging Community webpage of the Wellcome Center for Integrative Neuroimaging, because if you're doing MRI, I think the chances are very, very high that you're doing this at WIN to some degree. So join this community to have those conversations. For people who do lab biology, I've also posted a book there by Stan Lazic that I found super helpful and really interesting to think about experimental design. And I think there are resources out there. It's just sometimes quite hard to find. And like Guillaume said, maybe you just need to ask someone who knows about this. I'm also going to post a link to the BNA credibility website, because the British Neuroscience Association is trying to help you find the right resources for this too.
Yeah, it's difficult to give resources that would apply to everybody. There's plenty of resources for EEG and ERPs, but it'll be mostly in articles and so on. There's nothing yet that can give you a proper power analysis for ERP data. I've just submitted a grant application today with Cyril Pernet, where we're working on that, because you need to have a generative model, a physiological model of the data and how you get data on the scalp. And based on that, simulate effect sizes and run proper simulations. It's quite complex. So you can do surface things, like if you assume you measure just one component, it'd be like measuring one voxel in fMRI. But if you want to have something with a proper model, there's just nothing on the market. I know there's different groups working on it, but it is very complex.
Fantastic. So I'd like to go ahead and take a few audience questions now. I think it would be great to start with a very practical question that Lucas posted in the chat. I know you already addressed it in the chat, but Lucas, would you like to ask your question with your microphone and camera for everyone to see?
Sure. Hi, everyone. Great discussion. Yeah, I mostly asked a question to Guillaume because he said it's so important to share data. And for me, the question really remains, at which level should you start sharing your data? So my raw data are several terabytes. Then we pre-process, so it's mouse EEG and mouse EFIS. You pre-process it already at that stage. If you want to change something, for example, sleep scoring or so on, you could, in theory, tweak the data. And then you end up with huge data tables, which are sort of organized in a way that I think only our lab really understands it. And from there, at some point, you get to sort of the final data table from which you then do the statistics. So I feel if I only present, even if I present individual animals or individual data points, there's already such a long data processing pipeline before that there's a lot of sort of space for error or also for dishonesty.
Do you actually, I think it's important not just to share the data, but to share the code. So do you do that? So that somebody should be able to reproduce all your data pipeline, and that coding should be available.
Yeah. So I think we share the key code, but there's already so much pre-processing.
Yeah, but you need to share the pre-processing as well.
Yeah.
So that's the idea. So then you could put up, even if, and I often find that having the code available is more important than having all the data. I mean, the code with some sample data, so that you can actually work out what people did, and then you are reproducible, because somebody, as you say, this is the problem. This is what people talk about a lot, but it's only people in my lab that know exactly how it works. And what then often happens is that a key, I mean, there's this wonderful talk by Florian Markowitz about how then the key postdoc goes away and leaves the field or decides to stay around the world. Nobody can do anything with the data. You know, it's really serious, particularly when you've put in a huge amount of effort gathering these data sets, and you can end up being really horribly dependent on one or two people who will make mistakes. The other thing is we all make mistakes. So it's incredibly important to have every stage of the processing documented, whether it's pre-processing or subsequent. So to my mind, that's a real, that is another thing that takes time, but I think we're going to learn that you have to do it. It's very rewarding when you do, because you yourself will otherwise forget what you did. And so it's useful. You're partly doing it for your future self. And also I've had amazing emails from people who said, you know, how helpful it is for them. They may not be trying to check whether you did it right, but just to have somebody else's code to work through and see what they did. So often that's sort of hidden away and rather mysterious, and to have it out there is fantastic. And it really helps science progress.
Yeah, I mean, I can guarantee you save a huge amount of time. In the number of times I've been asked for, even, you know, in the lab, someone new starts and you want to give them a data set that's close to what they will be working on. On you go, you just send them the link, all the data and the code are there, they can start working on it. We
have a query from Alexander who says often the code is poorly written and is an archaeology expedition to get it running. Indeed.
So it's a bit of a trick there, because, you know, if you think your analyses are good enough to be submitted to a journal, then the code is part of the scholarship, is part of the work you've done. So if you can't share your code, then don't submit your paper.
Oh, you're a harsh man.
Well, I'm saying it that way because I've had it from a lot of authors
Yeah,
who are, they want to push their paper, but they don't want to have too much scrutiny. And I think, if your code is messy, well, messy code is better than no code, and just share it.
Can I also quickly respond to Lucas?
Absolutely.
Because I think it's a very interesting issue here. He talked about terabytes of data, of raw data. And I think the interesting question that I have is, how are those data kept and where? And is there a possibility to just share a link, right? Because, why would you have to share those data in a completely separate repository? Is there a way in which you can have a repository in your institute and in your lab that you can make publicly available for people to use, so that you don't have to store those data in two different places, one that's openly available and the other that's just available to lab members. Is there a way that, I think it's one of the nice things about the open science framework, if you use this for your data management, and I'm not suggesting you do this because terabytes of data is probably not what they can handle, but I think the nice thing about the open science framework is that they have this nice little button in the corner that says make publicly available. And so you can make whatever you want to share this at the stage when you're ready to share it. And then you don't have any additional work, right? So I think that's the point that I'm trying to make. Don't make this an additional step, the data sharing step, but can you have some sort of data management that allows you to do this, at a button click, basically.
That's the future. The future will be when, at the institution level, you are forced to format your data and describe them in a specific way. So you don't choose how you set up your folder, how you name your variables. This is forced on you. You don't have a choice. Otherwise, you're not allowed to do research. And then, as part of that, once you're done with your project, you can press on the link and it's publicly shared. And we have international standards for how things will be shared. And then we solve so many problems. These solutions are being worked on by a lot of people, but they don't exist yet. I think that's your point about having two systems. You have your own storage and then you need to turn it into something else. That's why we're wasting so much time. I mean, I'll be ashamed to show you how a lot of my projects I've managed over the years, it just looked like a mess. Since I've decided to share everything I do as much as I can, I've improved the way I organize my data a lot, but it's not there yet. But it would be dramatically better if someone said, "No, that's how it's going to be. Everything's going to be named in specific ways. You're going to have to structure that." So if you have EEG data, for instance, or MEG data or FMI data, you have the BITS format, where people have international experts have decided on the best way to structure the data. And that means the data can be read across lab because it's all organized in the same way. So you don't have your own particular techniques and descriptives fields and so on. So, but that's going to take time. That'd be fantastic. Imagine that.
Absolutely. And I see there's a lot of great discussion going on in the chat. And for any students and postdocs, we'd love to have you in the pub chat afterwards to continue this discussion. But for now, we're running low on time. And so I think we've got time for one more question from Antara about the interface between reproducibility in science and issues of diversity and inclusivity.
I'm very happy to take this because I feel very strongly about this. So I always say, when we're thinking about open research, open scholarship in any way, shape, or form, I think this has to go together with openness in terms of who we're open to and open for. So I'm a big fan of thinking about open research together with diversity, inclusivity, and equality. And I think this has to be an integral part of what we do. I think the great thing is in the UK, especially with Athena Swan initiatives, there are already systems in place to deal with that side of things. I'm not saying that these are perfect, far from it. But I think that's why in a lot of the initiatives that I've been working with, we haven't focused on that part as much as we as I would think we should, because this is part of the equality, diversity, inclusivity group, and they tend to be separate from the reproducibility communities in some ways, but they shouldn't be. It's one of the things that I'm trying to promote here in Germany now, that we think those two issues together, because I think we haven't focused on both as much as we should have, and I think it makes life much easier if you think them together.
Have you got any follow-up on that, Antara?
Oh yeah, sorry, I was just like, thank you for answering that question. It's mostly because I was concerned that this whole discussion we're having about reproducibility, it's, for me as a young scientist, I don't want to be caught up in this wave of like, this almost like culture in science where it's like people are trying to push out papers as quickly as possible. And I also feel for people who are in the position where they want to make their path in science, but they might already be facing a lot of other issues, like a lot of diversity issues and inclusivity issues. So how can they avoid being swept up in this wave? So thank you for answering like the question. And I know about the Athena Swan initiative, and I would like to hear more about other initiatives that you think can be put in place in the next 5 to 10 years even, by people who are leaders in the field to help propel the fields in the right direction and to encourage more people to go in the right direction for reproducibility. Thank you.
Yeah, I think all of this is around research culture, right? And the question how research culture needs to change. And this is how I see reproducibility as well. To me, this is just one sort of jigsaw piece. The jigsaw that we're looking at is really research culture and what would be the optimal research culture for the things that we want to achieve, right? Whatever that may be in your particular area of research. And so I think equality, diversity, inclusivity is part of that. Openness is part of that too. Reproducibility and replicability is part of that too. And so I think we all need to fit those pieces together. One of the things we haven't talked about today is, for example, bullying and harassment, right? These are also issues around research culture that desperately need to change for many of us. And I think that's also linked with issues of reproducibility and replicability. So I think we're facing incredibly many issues. There are great initiatives, for example, by the Wellcome Trust. The Wellcome Trust does incredibly many things on research culture now because they've realized that they have wasted money on irreproducible research, and that's partly to do with the way that research culture is not fit for purpose at the moment. So I think there are a number of funders now that are looking into this, and I have the impression things are improving. It's going to take time, yes, but we always have to have a long-term vision of where this is going.
Do you have anything you'd like to add to that, Guillaume?
Yeah, well, it's not being, being a white male, I don't think I'm the best person to talk about these things because I comes with all sort of privileges and biases, which I'm really aware of. The one thing I must say is just, even in my situation where I've never had really issues, and never had to fight for any of those things. Being part of that community pushing for open science makes me feel much better in my daily job. Quite often I felt over the years really isolated, and sometimes feeling like I could easily quit and go do something else because I feel like so much stuff is absolutely rotten. But being part of the community, especially on Twitter, is really great, and it's a really supportive environment. So I think that would help with other issues related to inclusivity in general for all sorts of reasons. Yeah.
I'll also add that for those of us who aren't as inclined to be very active on Twitter, there are other spaces growing in the online community as well. Like eLife community comes to mind immediately. And so I think that having your own sort of community is so important in combating the existing culture in academia and in research that is not always the most helpful. With that, oh, do you want to add a final note, Verena, and then we can wrap up
maybe
the
just
panel?
a final note. I love working with the eLife ambassador group. I think they're absolutely amazing. And one of the things that I've noticed over the last couple of years is that the community of people who are interested in culture change, in openness, in reproducibility, in diversity, we are a lot more diverse than the senior crowd that we are currently working with. And so I think it's a great space for people who care about these issues to get involved and find a community of like-minded people. I think we're that's one of the things I learned. You know, we are very open to different views, to different outlooks on the world, and I think it's so important to have those discussions, to have the views of people heard who may not sit around the table normally. And it's one of the reasons why I actually always say we need to have early career researchers in the room, right, where decisions are being made. We need to have much more discussions that include us, because we have a slightly different view of maybe how academia should be done, how diverse and inclusive our culture should be, because we don't have this survivor bias yet in the early career researcher group. So I think it's a great argument for why we should be part of that discussion too. And I really hope that we can convince senior people to let us be part of that discussion.
So this has been a fantastic discussion. I want to thank all of our panelists for taking the time to speak with us and for their wonderful advice and for the lively discussion from the audience in the Zoom chat. I've seen a lot of great resources posted. Alex mentioned the Growing Up in Science seminar series, and the Cortex Club's next seminar in this vein is going to be in early December, details forthcoming. And I would like all of you to give our panelists an applause using either the reaction button in Zoom, or if you want to unmute your microphone for a moment. And I hope to see you all at the pub chat, which will be in 15 minutes at 6:35. All right, thank you, and I'll see you all later.
Thank you so much, and see you later.
Thank you. You probably want to save the chat, right? Because there's a lot of resources in there.
Yeah, I don't actually know how to save the Zoom chat.
If you click on the 3 dots at the end, there's an option to save the chat as a text file.
Ah, fantastic. I'll do that, and I'll see if we can share this resource after the seminar.
That'd be great. Yeah. Okay, so see you in what, 15 minutes?
Yes, I'll see you all at 6:35.
All right, gonna grab a beer then.
See you.